in that case, hi. I'm Haley Branch. I am a postdoc at Yale with Jen Coughlin, but today I'm going to be talking about my PhD work at the University of British Columbia with Amy Engart, looking at rapid evolution to drought in Mimulus cardinalis. And for today, I'm going to be talking about these two overarching questions that I had, which is whether geographically distinct populations respond differently to extreme climatic events, and what the role of past climates have on the phenotypes that are expressed. And for the purposes of simplicity, I will be talking just about two traits. I did a lot of traits. If you are familiar with anything that I've done before, it's usually two mini traits. So today I'm just going to look at specific leaf area, which gives you a bit of a proximate for growth, so vegetation of the plant, and then flowering time, which gives you the other aspect, so the reproductive timing and the phenology of the plant. And during my PhD, I studied a not so recent drought in California that occurred, and at the time it was the most unprecedented drought, so the most severe drought that had occurred in over 1,000 years. Unfortunately, now we have seen that it's been worse, and we know that this is just going to continue to happen. And so I'm really interested in how plants respond and might either acclimate or adapt, either plastically or genetically to these types of events. And on the spectrum for plant adaptation to drought, there's kind of this broad spectrum where on one side there is drought escape, and this is kind of the live fast, die young strategy where plants grow really quickly. They're often more annual-like. They reproduce prior to the onset of an extreme drought. And then there's the avoidance side of this, where plants grow much slower, so they have a slower assimilation rate of carbon. They have less water use, and this is often through morphological modifications. I had the awesome opportunity to work with this phenomenal system, which is Mimulus cardinalis, the scarlet monkey flower. And what's really interesting with this system is that across its range, there is a wonderful climatic line here where in the north, it has much higher annual precipitation than in the south, and the variability in that precipitation also has this negative climb towards the south. And this really alters the demography of the plants and the morphology of them. So in the upper part, you can see the northern plants are much more dense, so there are more individuals, and the individuals themselves are much larger. Whereas in the south, which is indicated here by this one small plant, there's fewer individuals, and they tend to be quite small and short-lived. Prior to me arriving at my PhD lab, colleagues had gone into the field every year and collected these seeds, and so we had seeds already stored from prior to the drought and then throughout this drought period as well. And this allowed me to do a resurrection study where seeds from these time points, so descendants and ancestors of these populations, could be grown side by side in a greenhouse experiment. And in general, we're able to attribute any changes that are observed to genetic change, so evolutionary change. But that might not be entirely the case, and I will talk a bit more about that. So start with the first question about whether these geographically distinct areas resulted in differential evolutionary responses. I worked with Daniel Amstedt, who is a new PI at Cornell University, and we grew these plants in wet and dry treatments. And again, I'm going to focus on these two traits. So if we're thinking about escape and avoidance for specific leaf area, we have these really clear trends where on the escape side, plants tend to have broad, thin leaves. This means that the surface area of the leaf is quite large, so the solar radiation is higher, and that requires more evaporation, so more water use, and they tend to be very cheap leaves. On the avoidance side of this, you have the opposite where the leaves will be narrow and thicker. So they often have greater water use efficiency and they have reduced solar radiation, and therefore they can continue to assimilate carbon without too much water loss. And to walk you through what data I'm going to be showing you, I will always have avoidance and escape along the y-axis corresponding to the trait of interest. Sometimes the position of those will shift. And then along the x-axis is years. And so if there is a mean change of that trait, that would correspond to an evolutionary change. And then between these two different colors corresponding to wet and dry, that would indicate plasticity. And there's also a change of slope, so that's just the rate of change that I will also have on the other side of it, just to give you a clearer idea of what we're looking at. And so for specific leaf area, what we found was that the north didn't really have any clear trend, and specifically and most importantly in the dry treatments. Whereas in the south, we saw a loss of plasticity, and really importantly, the plants that were exposed to dry treatments, we could see that they evolved towards an avoidance. So they had thicker and thinner leaves. For date of flowering, when we think about this avoidance and escape, again, if it's escaping, it's going to be flowering quickly and then dying. Whereas we have the opposite if they're avoiding. And we saw a very similar trend as to the SLA. So in the north, we didn't see very much change. There was a little bit maybe towards avoidance, but in the south, we saw a great amount of change, and that's really highlighted in the rate of trait change from the slope graph you can see. And so the takeaway from this section is that the north changes very little, and whereas the south rapidly evolved towards avoidance. And some reason for this could be that the severity of the drought was higher in the south, or the length of the drought. And so that got me really interested in the role of kind of the past climates on the phenotypes. So during that drought itself, what might have been causing this difference. And so I then looked at 4 different metrics for climate that I thought would be corresponding to these changes. And here we have the first 3 are precipitation oriented, with the specificity of it increasing. So from the top is the most specific and then down, and then mean annual temperature. And I ran a bunch of models to see which would be the best kind of indicator of trait change, and it consistently came out as mean annual precipitation anomaly. I then looked at whether there was a difference with the current year. So a lot of the time in these types of studies, we look and we kind of map these traits onto their current year, but whether the previous year or 2 years prior would be more important, or a cumulative metric of these. And consistently, we had 2 years prior being the best climate variable year to describe trait change. And so I'm going to present these graphs here, where on the x-axis, the negative numbers correspond to drought years. And this is for specific leaf area. So this is just for the north. And what I really want you to take away from this is that the trend that you can observe is very different when it's the current year versus 2 years prior. And the current year actually wasn't even selected as being an indicator for trait change. So 2 years prior was a better indicator of this. And so this could be evidence of some type of fluctuating selection or something conflicting. For flowering time, we saw the exact same thing happening. And so again, this kind of made me start to think that maybe that's why we didn't see a clear trend when we just looked at trait change. But when we look at the climatic variables here, there's clearly something happening that's altering, flip-flopping for the plant. Whereas in the south, there wasn't a clear indicator of which year. And what we actually found was that for both specific leaf area and for flowering time, that it was just consistent, which is also consistent with just the longevity of that drought and how it was pretty much stable that whole time. And so it was a longer press perturbation. And so the takeaway from this is that 2 years prior was often better explanatory for trait change in the north, and that this conflicts, and that in the south that they were equal and the same direction. But then it got me thinking about why this was only happening in the north and what could be different. And just looking at this little schematic of going back in time, it got me thinking about transgenerational plasticity. And so whether grandparent to parent to offspring was actually a big contributor to this. And so you're probably very familiar with epigenetics, but just a quick brief. When we're talking about transgenerational plasticity, this is when the grandparent has experienced a stress, which then will alter the way that the DNA is then transcribed and expressed. But then following 1 year, so the parental generation doesn't experience the stress, but then the offspring also doesn't, but still expresses the grandparental phenotype. And the places that we're most likely to see this is when environments are less variable. And so this is where we expect this to evolve and be potentially adaptive. Also in areas where where there's not as much genetic variation could possibly be where this is, and where it might be important to be highly responsive or have really quick generation times. And so what I then did was I looked at the interannual precipitation variability, and so how predictable was precipitation from year to year. And I found that historically in the south, so that's indicated by the orange, it had the highest variability, whereas the south did not. And then during the drought period, the south consistently was the same, whereas the north became a lot more variable. And so quickly, the next data I'm going to show you looks like this with these violin plots, and along the x-axis is treatment history. And so each one corresponds to the grandparent, parent, and then the current year, so the offspring. And so if there's differences between these two groups, it's parentally derived, grandparentally derived, or both. And so what we found was that for the north, there was actually a lot of evidence that when the grandparent had experienced a drought, that it resulted in an escape strategy. And this was also seemed to be cumulative in nature, so where it was more significant the more drought history there had been versus how much wet history there had been. Whereas in the south, we didn't see any evidence that there was transgenerational plasticity. For flowering time, we had a similar trend where if both the grandparent and the parent had experienced drought, we had the earliest flowering, so the most likely to escape drought, versus when there was 2 prior generations of wet. And again, in the south, we did not see any evidence of this. And so I then quickly did an analysis to look at the effect size of whether transgenerational plasticity or just year, so we attribute that to genetic change, and which was more corresponding to trait change in the north and the south. And you can see that in general, the north transgenerational plasticity was more important than genetic, whereas in the south it was the opposite. And so overall, this gets at why the north might not have actually observed much change overall. So we have this conflicting potential fluctuating selection that is also associated with transgenerational plasticity, which would then kind of feed back into very little change being observed, whereas the south had a significant amount of change towards avoidance and a continuous press perturbation and no real transgenerational plasticity. And so with that, I'd like to thank you all for coming to my talk, and I'm happy to take probably one question. Yeah.
Great talk. I have clarification. Why is it, couldn't it be, you're calling it plasticity going from the grandparent to the offspring, couldn't it be a shift in the gene pool, which would be a genetic explanation? So if you can help me understand.
Yeah, so I didn't get to fully explain, I guess, the methodology behind this. So I grew up these plants and actually exposed them to themselves. So this wasn't in the natural environment. So I exposed these plants, which we had already done a experimental evolution study with. So these plants, I selfed them, and they went through each iteration of these drought. And so any change should be more related to, and especially since we can see these differences across each group, it's less likely that it would be attributed to genetic change. But I am doing methyl-seq on the data right now, so we'll get more information from that. Thank you.
Hi, everyone. My name is Fouilloux, and I'm a postdoctoral researcher at the University of Wisconsin-Madison, and I'm working in the Height Lab with a lot of other cool people. And today I'm going to talk to you guys about antimicrobial resistance, or AMR. And that's becoming an increasingly concerning global health problem. And it's been a pretty hot topic for about a decade because the idea that drugs are no longer going to be effective at combating infection is like a pretty scary reality to deal with. And so as scientists, we've done a really great job to go around the world looking for these clinically relevant resistance genes. And we've found them pretty much everywhere. We see clinically resistant strains in Svalbard, where there's more polar bears than people. And we also see these hot spots in places that we presume are affected by inappropriate waste management strategies, which is going to be increasing your antimicrobial exposure risk. But the question in all of this is, do AMR genes in the environment have consequences for public health and the wild animals whose microbiomes that they co-opt? And so up until now, we've done a really great job at documenting resistance and documenting hotspots, but we don't know the consequences that AMR has for animals and the natural populations they're affecting, or for public health consequences. And fundamentally, we also can't really explain this map in the sense that we can't explain why there's more AMR in some locations compared to others. And that's because AMR is really complicated. It results from a complex interaction between the host, the environment, and other stressors. And so we have a really limited understanding of the main drivers of AMR, especially relative to the role of environmental pathways. And so really what's been missing in this story is moving beyond documentation and integrating like this host, genetic and environmental information to allow us to actually predict antimicrobial resistance dynamics and prevent outbreaks in these real dynamic systems. And so I'm really, really lucky to be able to kind of have an amazing case study to talk to you guys about today. And so I've had the enormous luck to both contribute and work on a data set that spans from the antimicrobial resistance gene level that scales all the way up to host and environmental landscape data. And so this is pretty much a gold mine to be able to develop a framework that's connecting these pieces of the puzzle that we need so badly in order to think about AMR from a mechanistic perspective. So, welcome to Vancouver Island. Vancouver is home to the highest density of commercial fisheries in Canada. And so today I'm going to tell you about my work that's built around the ecology and genetics of a really well-studied fish, the wild stickleback, you probably heard from him today, from 47 lakes that span this entire island to understand the drivers of antimicrobial resistance in these wild populations. And so we've got the aim in progress of eventually identifying the relative contribution of different drivers to understand what's actually driving AMR through structural equation modeling. So if we imagine we've got AMR genes and we've got two specific targeted genes, and these genes exist within a host. The host in our case being these stickleback fish, and these fish exist within these lakes, and so we've got this host information, sex and length, we've got this limnological information from the chemical and physical profiles of these lakes, and there's also the way that these lakes are being impacted by the environment from environmental drivers like runoff and deforestation and agriculture and aquaculture. So all of this is coming together, and our ultimate goal is to build this model, and that is in progress. But today what I'm going to be showing you are some really important patterns that I've identified. And so just to touch on the genes, when I say AMR genes, the ones that we're going to be talking about today are two specifically that we've sequenced already from gill swabs from these stickleback, both of which are associated with aquaculture. So we have FLOR, which is a phenol resistance gene, which is the most common antibiotic used in fisheries. And then we've got TAP, which is a pretty nasty tetracycline gene, and that's used as a growth promoter in a lot of stock animals. So I'm going to start with all of the results now, because we all have things to do. Across the 47 lakes, we sampled many, many, many fish and find a pronounced spatial variation across the island, kind of the same as the map I showed you guys earlier. And so we have that same fundamental question, right? Like, why? Why is there variation in these lakes? So I'm going to be spending the rest of my talk approaching these from 3 different hypotheses and thinking if we can start to identify any patterns and why they might be. So to start with kind of the most intuitive hypothesis, is that point sources drive antimicrobial resistance. So unsurprisingly, you might imagine that these salmon fisheries, they're spilling over into the surrounding environment. And so we would predict that the closer that a lake is and fish are to salmon fisheries, the higher probability that you're going to have antimicrobial resistance in those fish. So let's look at some field data. When we actually look at this, we actually see that our data corroborate this pattern, but there's a lot of variation there, right? And so this isn't surprising considering the heterogeneity of these spatial data. I mean, it's 47 different lakes, and so if we had one variable that captured all of that, it'd be pretty amazing. So we've got also other hypotheses to think about, and we can really think about how habitats mediate AMR inputs in the sense that not all lakes are created equal, and they're going to be dealing with these inputs quite differently. And so if we think about this in the context of runoff, we have these point sources that can be, from agriculture or different land practices, and these habitats are then subject to the pressures, and some of them are going to be unable to mediate the consequences of these inputs, and they're going to become eutrophic. And so we predict that eutrophic habitats will have higher AMR, in part because they're already very clearly affected by some environmental change. And so when we think about eutrophic lakes, we're going to be thinking about lakes that are relatively higher productivity, higher dissolved organic matter, lower O2, and lower pH. So let's go through some findings. So when we think about productivity, we're thinking about a strong positive correlation, right? So higher chlorophyll, higher AMR. Our field data seem to corroborate this pattern. For those eutrophic lakes, when we think about dissolved oxygen, we're going to think in more anoxic conditions, you're going to have higher AMR. Our field data corroborate that pattern. I think you're sensing where I'm going with this. In lakes that are going to be more acidic, we would also imagine higher AMR, so again, that negative correlation. Our field data corroborate that pattern. So I think you're probably getting the download here, that these habitat qualities really appear to come together to paint a story about eutrophic lakes. And it highlights that what these systems receive as input and what survives and thrives in certain habitats appears to have important consequences for the prevalence of resistance genes in these lakes. And I think that's a really great way to think about runoff and the habitats, but we can't forget the stars of the show here. We still have hosts that have these AMR resistance genes. And I think it's been pretty well covered throughout this conference already that the microbial community of a host can be drastically different from the average microbial community of the environment that this host is found within. And that's really important for us because animals can shift their space use depending on diverse factors such as seasonality. And so when we consider this in the context of stickleback biology, June actually coincides with stickleback breeding season. And so, and this is when we sampled all the fish. And so the common stickleback, they actually build these pretty cool nests. These males make these nests out of algae and soil that the females come and visit. And so this is really important because genes are most often associated with bacteria, and these bacteria are then colonizing specific microhabitats. And so, for example, the phenol resistance gene appears tightly related to Mesorhizobium, and Mesorhizobium are a genus of soil bacteria. So if we were predicting if males or females are going to have more AMR, we would expect that males would have more AMR because they're in the soil, building these nests, attracting their ladies over. And so when we look at this, compared to our field data, we actually see the exact opposite trend when we go for size-corrected AMR rates, which is exactly the opposite pattern I would have expected. And so this is really important when we start to think about host biology because perhaps we're seeing higher rates in females because of that reproductive stress, because it's the breeding season. If we put this in the context of other immature females, again, the data here are sparse because we were collecting during the breeding season, but when we compare the AMR rates in reproductively mature females compared to females that are immature, we're seeing significantly higher rates of AMR. And so in this context, we can actually leverage epidemiological modeling, and we can think of this from a disease standpoint in the sense of the acquisition, let's say, of AMR is almost like the transmission of a disease. So if we think about the susceptibility, perhaps these females have decreased immune function because they're diverting that energy to reproductive efforts. And then if you think about exposure, well, they're going down to visit these nests that these males are making and, getting lost in that bacterial sauce. Maybe that could be an example of why we're seeing those higher rates in females. So what's this story telling us? Well, if we want to move beyond documentation into predicting and preventing AMR outbreaks, we need to consider multiple drivers, which is no easy feat. We've got this idea of these point sources that come from these diverse places that are being affected in different habitats, and then we've got these players that are then, different ecomorphs or phenotypes that are then existing within these habitats in different ways. So the idea that AMR bacteria exist everywhere and can jump ship at any time is pretty scary. And so I've told you today that by accounting for these interacting factors, we can move beyond documenting hotspots towards a mechanistic understanding of what drives AMR in natural populations. That's really the first step to understanding the consequences for both natural wild systems and public health, and improving our ability to both predict and prevent future outbreaks. So with that, I'd like to thank so much the Height Lab, that is the UW, come say hi to us, and then also the Bolnick Lab and a lot of cool people down at Carleton, and you for coming here after the lunch. Thanks. Not all at once. Yeah.
Do you know, for your resistance genes, are those living on the chromosome of the bacteria or in plasmids or something else? Or?
Yeah, so we've got those mesorhizobia that I showed you seem chromosomally associated, but that tap resistance gene seems to be on a mobile genetic element. So I think that one's especially spooky because I think that'll be able to really horizontally transfer to other bacteria too. Thanks. All right.
Chloe, and other systems do they know about variation in males and females driving different patterns of molecular resistance, or why reproductive stress might lead to a stress response in the host, then leads to a stress response in the bacteria that elicits these AMR genes, or?
Yeah, so from what I understand already, you've got different rates of how susceptible they are based on how they're navigating the environment. But if there's a sex-specific reason if they're more susceptible to AMR, just physiologically, if we don't think about immune, but just pure susceptibility of what your microbiome is, I'm not sure, no.